Google DeepMind's AlphaFold, unlocking the power of AI in science. 5 years after its debut, Google DeepMind's AlphaFold has revolutionized the field of protein structure prediction, showcasing why science is AI's killer app. John Jumper, a senior scientist at Google DeepMind, expressed his amazement at the technology's impact, stating that it exceeded all expectations. In 2024, Jumper and Demis Hassabis, co-founder and CEO of Google DeepMind, were awarded the Nobel Prize for Chemistry for their groundbreaking work on AlphaFold 2. AlphaFold 2 has become an essential tool for graduate-level biology students worldwide, teaching them how to use it for protein structure predictions. This technology has transformed the way scientists approach the complex world of proteins, which are the engines of life, controlling various biological processes. Prior to AlphaFold, determining protein structures required time-consuming and expensive lab experiments, limiting the number of known structures to around 180,000. Google DeepMind's innovative approach involved using a transformer, similar to the AI powering popular chatbots like ChatGPT. However, instead of generating text, the model was trained on protein DNA sequences, known structures, and evolutionary clues. This enabled AlphaFold to predict protein structures with remarkable accuracy, leading to the identification of over 240 million proteins, including those involved in critical human diseases like COVID, malaria, and Chagas disease. The company made AlphaFold 2 freely accessible to researchers, providing an internet-based server for DNA sequence uploads and structure predictions. The European Molecular Biological Laboratories European Bioinformatics Institute has deposited these predictions in a comprehensive database. AlphaFold's impact is evident in over 40,000 academic papers, with 30% focusing on disease research, and it has contributed to approximately 200,000 research publications. One of AlphaFold's most remarkable applications is its role in uncovering life's mysteries. For instance, scientists used AlphaFold to discover a previously unknown protein complex essential for sperm fertilization. Andrea Pauly, a biochemist, praised AlphaFold's speed in accelerating discoveries, making it a vital tool for her team's projects. AlphaFold has also played a crucial role in understanding heart disease and honeybee populations. By predicting the structure of ApoB100, a key protein in LDL cholesterol, scientists gained insights into its complex interactions, potentially leading to better treatments for high cholesterol. Additionally, AlphaFold helped identify the structure of vitellogenin, a protein vital to honeybees' immune system, offering hope for enhancing disease resistance in these pollinators. The accuracy of AlphaFold's predictions varies, with a confidence score indicating trustworthiness. While some proteins have inherently disordered regions, AlphaFold 3, a more advanced model, can predict their binding behavior with other proteins or molecules. However, the impact of AlphaFold on drug discovery is still being assessed, with ongoing research exploring its potential in repurposing existing drugs and designing novel ones. Google DeepMind's sister company, Isomorphic, is at the forefront of drug design using AlphaFold 3 and other tools. Partnerships with pharmaceutical giants like Novartis and Eli Lilly indicate the technology's potential in the industry. Additionally, AlphaProteo and AlphaMessense, created by Google DeepMind, showcase the AI lab's commitment to advancing protein design and disease understanding. Looking ahead, Jumper expresses interest in exploring the role of large language models in science, envisioning them as powerful tools for hypothesis generation and experimental design. While some AI startups have experimented with these models, Jumper remains skeptical about their effectiveness in designing novel proteins. Nonetheless, the future of AI in science looks promising, with AlphaFold and its successors set to unlock new frontiers in research and medicine.